That's good for me. Okay, just some.
Okay, we can see your screen. You can go on presentation.
Issue. Yes, I'm trying to go there.
In the meanwhile, Professor Banni has been one of the main speakers in one big conference that we are organizing with the Metabolism of Cities Living Lab together with other organization. That's in the International One Health Conference, in which we are trying properly to address the nexus between health and environment. And he has been, okay, here we are, we can see.
Here we are, okay. Good. So just a brief presentation of my status, Professor Mohammed Banni. I'm leading a research group on ecotoxicological aspects in relation to the effect of contaminant, the mainly emergent one, on cellular function and molecular process. I mean, we are trying to establish and to make the link between the environment, its components, its hazards, and the human health. I'm professor actually of molecular toxicology at the Higher Institute of Biotechnology of Monastir and leading a research group in my laboratory called Agrobiodiversity and Ecotoxicology. I'm also invited professor in some prestigious European universities, such as the University of Vanvitelli in Napoli and also in Paris, University UPEC. I'm also until now, I have supervised 22 PhD theses, PhD program, and published around 130 scientific publications. Yes, also I'm leading some European project from the bilateral, collaboration between South and North part of the Mediterranean, including Horizon Europe, NECBC Med, and Cross-Border and Prima projects. Yes, that's it. So our presentation will be around microplastic, the One Health concept, and I will try to present this so-called interaction between microplastic from where it reached the environment, and then how could this environmental contamination impact at the very important endpoint, human health. So I will start with this usual and also very important slide, which represent the main sustainable goals, development goals, as established by the European Commission. And microplastic can be associated to many of them. For example, the first one, which will be good health and well-being, the third SDG, or Sustainable Development Goal. Microplastic is in the heart of the health, human health and well-being, since we reported recently a lot of interaction of microplastic and human health. The second very important SDG will be quality education. Why quality education? Because how to combat, how to struggle against microplastic and plastic contamination. I'm more than convinced after quite 15 years of research that the only path that we should follow to contrast microplastic and plastic in general will be through the education, and starting from elementary schools. We have to implement the education and the good reflex, let's say, respect to the use of plastic, and this should start from really the early beginning of education. Clean water and sanitation, SDG6. Also here, we have a crucial problem around the world with the contamination of water that we drink, but also water in what we call the surface with water, that could be used further either for irrigation of soils and for agriculture, or directly to be drank by humans, or also by livestock. So the water and the clean water is one of the main aspects that we are facing now with respect to the contamination of microplastic. One of the also main SDG is the 13th one, the climate action. There is a direct link between the climate change at the global level with the increase of plastic contamination, since mainly in agriculture we are opting toward the intensification of the use of plastic, as if in greenhouses, in mulching, I mean covering soil to avoid the weeds presence in agriculture soil. And this is an indirect consequence of the climate change. Also, you can consider, I mean, one of the most famous recent episode that humanity faced is the COVID. And the COVID, unfortunately, increasing drastically the use of plastic and further the production of microplastic through the use of masks. I mean, masks and any other option that we use it to face in somehow COVID episode was also impacting the presence of plastic, and data are really incredibly indicating this. Then the life below water. Also here we have a very drastic problem with microplastic and plastic in our oceans, and the Mediterranean is one of the most affected, the most impacted seas in the world by the presence of plastic, since it received the water flow of many rivers that are carrying a huge amount of plastic. And recent data on the quantification of plastic reaching the Mediterranean, mainly from the big rivers in Europe, are really, are really indicating a very crucial problem. That's the first point: plastic and sustainable development goals. Then the second one would be this so beautiful illustration of the healthy people, healthy environment, and healthy animals, and how you will see through my presentation that the One Health concept is in the core of this presentation, and this should be also in all other aspects related to environmental contamination, with a direct impact of human health. So humans are impacting the environment. The environment is also making a pressure on living animals, I mean living livestock, for example, but also wild animals, and the the contamination of these livestock or of wild animals will impact directly the human health. One of the crucial problems now, probably not related to microplastic, but to other contaminants or other biological, let's say, diseases, is related to the West Nile virus, for example. Climate change, which are one of the main drivers of the biodiversity disorder, are now presenting some symptoms of contamination in Europe. Now, West Nile virus is more related to the southern part of the Mediterranean. Now, with the climate change, we can see them in the north of the Mediterranean. Ishmania also, and Ishmaniose was also reported in some region of North Mediterranean countries and cities. And then our relation with animals is not the same in Europe as in the South Mediterranean country. But now, with the Ishmania's presence in the north part of the Mediterranean, the relationship with dogs is more, let's say, friendly in this country, in this region, and then the transmission to human would be very, very crucial, and we are facing a real problem now. So we have to take care of our environment to avoid contamination of our animals, to avoid at the end diseases that could impact humans. This is the global idea. Okay, So why microplastics? Why we are dealing with plastic today? Because plastic was reported to be one of the main emerging pollutants in our day-to-day reflex. Plastic was reported in scientific reports, which are one of the most important, let's say, journal in dealing with the scientific aspects. Microplastic fragment and microbed in digestive tract of planktivorous fish from urban coastal waters. The plastic was also reported in commercial salt from different countries. So when you are using salt in your kitchen to cook flavored and good meal, you are introducing into your, let's say, plate some microplastic. And this will be driven would be in relation with a lot of problem linked to human health. Water, the water that we are drinking in our plastic bottles may contain, and I'm sorry to introduce this information, but for sure it contains microplastic. And this was a terrible, information that have been vehiculated with our friends from that have published it for the first time, this publication. This publication was also reconforted and strengthened by another publication of the group of Professor Margherita Ferranti from Catania, insisting of the presence of microplastic in water that we can use into bottles. So analysis of microplastic in water by macro-Raman, which is a very crucial technique in this kind of situation, release plastic particle. There is another very interesting publication that has been released by a group from UK saying that million of microplastic in a cup of tea. You know, when you are making your infusion cup of tea, you are also introducing microplastic, since the plastic bags that are used for the infusion tea may release million of microplastic in a cup of tea. And I advise to reach this publication. It is available and free online. Just put microplastic and cup of tea, okay? Then these are, now we are in our, let's say, lab evidence, and these pictures were taken in my lab in the frame of the Euro-Mediterranean project called Common. And here, as you can see, we have animal tissues. In this case, we are dealing with fish tissues, and we have identified microplastic sized between 10 and 20 micrometer in fish tissues. And then you can see by the micro-Raman analysis the color, the type of microplastic, and also the identification of the chemical characteristic corresponding to this microplastic. We are dealing with HDPA, high density polyethylene, with nylon, with polypropylene, and so on. So these are real pictures showing the presence of microplastic. And as you can see, I'm trying to start this presentation with evidence, just to show you that we are talking about something real, something that is really happening today.
Okay. Mohammed, sorry.
Yes,
sorry, I want to ask one thing. So you say that you use micro-Raman spectroscopy. I tell because it is a technique that also we saw into the previous lesson, and I was used properly to define which type of microplastic it was, no?
Yes. Micro-Raman is a technique that will identify on specific filter the first, because you are using specific filter with specific porosity, so you know more or less the dimension of your microplastic. But the micro-Raman will identify through laser the type of microplastic. And as you can see here, for example, you have a standard that indicate the type of microplastic that will be compared of the one you are identifying. So it will be, the micro-Raman will give you as main result this, let's say, chromatogram. It's not really a chromatogram, but this profile that will be compared with a database that you have in your lab to identify the type of microplastic. So the micro-Raman is mainly to identify microplastic type, chemical type, polyethylene, polyethylene terephthalate, high density polyethylene, and so on, PVC, and so on. So we have the database, and we have just to compare. Is it okay as explanation?
Yes, thank you.
Thank you. Okay, so here I'm dealing with this also with this illustration just to show you that these are real data that have been published recently from my group in liver and in seawater of liver of not dysentery of a wild species called Serrano scriba. It is a fish with a very limited area of, let's say, movement. So he can indicate the contamination of this site where it has been captured. And also in the same time, we are filtering seawater to see the presence of the microplastic. So here you have the two kind of approach, the presence of microplastic in seawater, and in the same time, the presence of microplastic in fish liver. So this is now another technique that we employ in the lab to identify microplastic. If this one, the first one, is a chemical-based analysis after digestion of the tissue, after filtration of the tissue, and then identification with microramen on specific filter, this is directly on the tissue without making any homogenization, nothing. I mean, you will take your tissue, you will make section of 5 micrometer sections, and then you will, let's say, make the dye with hematoxylin, eosine, and you will have a double coloration. You mean under normal light and under polarized light. Under normal light, you will see nothing. But if you switch to polarized light, for example, this is a polarized light. And under polarized light, because the polarization will give you an autofluorescence of the material, which is in this case microplastic. And in this case, it was a lab contamination. So we know very well that this is microplastic. The same here. These are gills. Here we are talking about liver, and these are the main blood veiculating, let's say, how to call them? Yes, vessels. And here you have the microplastic that are really very impressive and how they are reaching this region. Here you have something in liver, but from wild animals, and as you can see, these are microplastic that have been confirmed further with the Raman microscopy analysis using the other technique. So, as you can see, microplastic is not really a myth. No, it is a reality. So let's go to this really very important slide that will give you an idea about microplastic are toxic because of their physicochemical characteristic, or because also, and I will underline this word also, because also of the additives that can also be linked to microplastic. So you have two kinds of additives. The additives that have been used to produce the plastic itself, and we call them the plastifiers. The additives that are used to as stabilizers, but also you will have some additives that will adhere to the plastic in the environment. I mean, in the environment, while the process of aging, and I would also explain this word aging, because when you throw out a bottle of water in the sea, it will start the process of aging, the process of maturation. And this process is accompanied with an accumulation of chemicals from the environment into the plastic. And these are called also additives. I mean, substances that could adhere to the plastic in the environment, but not only chemical pollutants such as PAH, PCB, that could be also metals, pesticides, phthalates, that could be also some PFAS, but also microorganisms can adhere to the plastic, forming what we call a biofilm. And this is a crucial point because some bacteria could not survive in seawater, but if they will found somehow what we call an ecological niche being in addition to the plastic, they could survive. And this is a crucial point when we are talking about pathogenic bacteria such as E. coli or such as also Vibrio fischeri and so on. So plastic would provide a survival environment for this pathogenic bacteria in marine environment. And we found this and published this, and I will arrive to this point. So this is a very important point. Are plastic toxic because their intrinsic properties as chemical properties, or because of the adsorbed substances that could be from the environment? And here we are talking about a chemical cocktail, because plastic is offering for us scientists the opportunity to study the mixture of pollutants that could be present on the surface of plastic. Here we can introduce a new scientific word called the Trojan horse effect. And I think that you remember the story of the Trojan horse and how this horse has been used to introduce soldiers inside the so-called fortress. So plastic can also vehiculate and introduce chemicals into cells. And this has been challenged, has been demonstrated, and I will provide some proofs. And I will begin with the first example of this presentation, which will be dealing with agriculture soils. So my three examples during this presentation would be agriculture and how we can fund microplastic into agriculture soils, and then probably you will find them the so fantastic sentence from the ground to the plate, but also how can microplastic change some fundamental behavior of key bacteria in agriculture soils. Okay, so we will deal with this in this first presentation. And this first example, we conducted a co-exposure to environmental microplastic and a pesticide, the first one in the world as use, I mean, in terms of amount of use of pesticide. It is an herbicide, the 2,4-D. It is an oxenic substance that is widely used in the world to eliminate undesirable plants, I mean weeds. And in this presentation, in this work, we have challenged agricultural soil with the herbicide alone, with the microplastic alone, with the mixture of them, and then we have studied the impact on the earthworm. Why the earthworm? The earthworm, Eisenia andrae, is one of the key factors that will provide some improvement into agriculture soils. Since it will aerate the soil, it will also act to provide this combination of beneficial bacteria and so on. So our focus was not the worm itself, but the gut of the worm. That's why we have dissected out the gut of the worm, and we have evaluated two parameters, in fact, three parameters. The first was the accumulation of microplastic and the herbicide into the worm. The second, the bacterial communities inside the gut of the bacteria using metagenomic approach, and the third, metabolomic approach using a quadruple TOF analysis of the main metabolites in the gut. And we tried to combine all these three parameters to understand wherever microplastic can increase or provide a synergic effect on the first effect of the herbicide usually present in our agricultural soils. So yes, the metagenomic analysis was conducted after extraction of DNA from the gut, then the amplification of specific target of bacteria, then DNA amplification and Illumina high throughput sequencing, and then the obtention of the sequencing and the enrichment. In the meantime, we did metabolomic analysis using an hybrid quadrupole Orbitrap mass spectrometer coupled with specific, heated electro spray ionization, and we obtained this so-called main metabolites in the gut of the worms. Yes.
Sorry, Mohammed,
Yes.
just want to summarize and eventually ask if there are some questions. So in the
Okay.
analysis, they take the worm as a sample. So they analyze the presence of microplastic, but also we can say the metabolomics in order to
Yeah.
assess the toxicology, no, of the
Yeah. Yeah.
of
Yeah.
the
Absolutely.
yeah.
We
Okay.
did not only analyze the presence of the herbicide or the microplastic, but also the diversity in the bacterial community inside the gut. So the effect of both contaminants on bacterial diversity in the worm gut, and then the main metabolites that the worm produces to face this situation. So it is such a kind of holistic approach. We try to understand what is really happening in the gut of these animals, since the gut in general is the equivalent of our intestine. And the intestine for a human is called also the second brain, because a lot of disease are provoked by an unbalanced situation in our intestine. So it is the same also for earthworms. And if the unbalanced situation happens for the worms, they would not be able to maintain their crucial, let's say, role into an agriculture soil. So this will lead to a great dysfunction in agriculture soil, and with further, consequence on the yields of plants and loss of yields for our farmers. So this is the intention. Okay, I'll go further. So the first result is regarding the presence of microplastic, and as you can see, microplastic in organism that has been challenged with microplastic is very high. But and we tried to work within 14 days of exposure, and we found out that in animal exposed to microplastic, animals can concentrate microplastic mainly with dimension that range from 1.22 to 0.45. So the so-called small microplastic. Probably I didn't mention this. The dimension of plastic will give us three types of plastic: the macroplastic, the mesoplastic, and the microplastic. So macro, meso, and micro. Microplastic started from 5 millimeters down. So 5 millimeters are the maximum of the microplastic range size. And we can speak here about big microplastic and small microplastic. Small microplastic are considered small if they are under 10 micrometers. Why this classification? Because microplastic under 10 micrometers can enter the cells and can provoke some alteration inside the cells. And we will speak about nanoplastic if their dimension is less than 100 nanometers, so 0.1 micrometer. Okay, so here we are dealing with a particle of plastic with dimension between 122 and 0.45 micrometer. Okay, and as you can see, in animal challenged with microplastic, the total presence of microplastic is huge respect to the control situation. Okay, and even in the control situation, when you found out something like 6.77 microplastic per gram of tissue, that means that also in control soil we have a minimum amount of microplastic. So we have no more the so-called plastic-free soils, if you want. The contamination is almost spread with different level, but it is there. So then we try to identify the type of microplastic with PA, PEVA, polypropyl, polyethylene terephthalate, and polyethynyl. And this is just for us an information to know which will be the origin of this microplastic. Then the second data, very important for us, is the accumulation of the herbicide inside the animals. And as you can see, in animals challenged with only herbicide, we have reached something like 0.5 45 milligram per gram of tissue. Sorry, here it's microgram per gram of tissue. It is an error. But in the mixture, we have something that around 0.59. And as you can see here, unfortunately, not unfortunately, but there is no significant difference between 2,4-D and the mixture with microplastic. That means that in this case, microplastic didn't significantly influence the uptake of the herbicide. But in other experiments performed in my lab, we demonstrated that microplastic can affect and increase the uptake of chemicals. So now we will go to the metagenomic analysis, but just to avoid the confusion with you, I will be very simple. So please don't consider this very complicated images and so on. I will be very simple in the analysis. So for the first time,
sorry, Mohammed,
yes?
just 1 second just to make a step on the previous slide. So they analyze the presence of herbicides because, as Mohammed has said, sometimes the microplastic can increase the presence of certain chemicals. No, we saw with the BPAs last time. No, they can catalyze the presence of certain chemicals. Thank you.
Absolutely,
sorry.
Yes. And just to be in line with what just have been said now, in other publication, we demonstrated that, for example, benzoypyrin was vehiculated by the microplastic. The key factor that will, let's say, make robustness this so-called Trojan horse effect is the chemical properties of the 2,4-D. I mean, the hydrophobicity or lipophobicity. It is very important to be associated to plastic. Anyway, so let's now make a focus on the metagenomic analysis. So for those who didn't deal with metagenomic, what is metagenomic? Metagenomic is an approach where you will extract DNA from a population of bacteria and then make an amplification of specific sequence that is common for all the bacteria, but not common in terms of sequence, but they are present in all bacteria. And then you will try to find out which are the main families, the main, let's say, groups of bacteria that are present in this population. So this is called metagenomic, and you can either do some sequencing of specific sequence or full sequence, and in the full sequence it is more robust and more specific. So here we found out for the first time the presence of archaebacteria in the gut of worm. This is the first report worldwide on the presence of archaebacteria in earthworm, and this really was something very welcome for us because we reported for the first time the presence of archaebacteria in earthworm, in control, but also in other situations. So the main data, I'll go directly to the main data. Yes, this one. No, this is the metabolomic. Yes, this the main data of our research regarding metagenomic. In fact, there is big difference between control, animal exposed to 2,4-D, to microplastic, and to the mixture. And the main family are listed here, nitrososphaera, for example, that are implicated in the cycle of nitrogen. And these are crucial bacteria. You have also the bacteria that are implicated in the cycle of carbon, in the cycle of sulfate, and so on. So the main chemical classes that are present in soil as mineral have to follow this so-called geobiological cycle. And this geobiological cycle is assumed and is due to the presence of specific bacteria. So we need them in the soil. And as you can see here from this graph, there is a big difference in their abundance respect to the contamination. So the contamination affected drastically the presence and the distribution of bacteria classes in the gut of worms. Second, this is the metabolomic analysis. And as you can see, the main metabolites that we found are more than 1,150 compounds. And all these metabolites have been ranked here through a heat map to show that, for example, from control, those exposed to 2,4-D microplastic and mixture are not the same metabolites. So we have different class of bacteria respect to the condition and different class of metabolites respect to the condition. So the data here becomes very complicated to analyze, but we need to find out a solution. The solution to make easier the analysis of this data is to make a certain kind of PCI and to make this so-called principal component analysis that will discriminate very good this population along with the metabolites. So as you can see here, the effect of microplastic alone, the effect of the herbicide, and the effect of the mixture are very different, one from the other, and all of them from the control, and all of them from the control situation. Okay?
Just Mohammed, one second.
Yes.
Regarding this technique that's called principal component analysis,
Yes,
we will not
statistical
go.
technique.
Is a statistical technique that basically discriminate the main prominent effect over, we can say, a sample of data. Okay.
Yeah, absolutely. Yes. I'm sorry. Probably I'm introducing some terms that you are not very familiar with, but hopefully my friend is here to make some adjustments and to make it easier to understand these graphs. Yes, this is the enrichment statistic plot for earthworms. In fact, here we are combining metabolomic with metagenomic. I would not really be very late here, but yes, here it is very important. So among the 1,150 components, we have created this small group that are driven by microplastic and the mixture of microplastic. I mean, the creatinine, the nicotinamide, the isonic acid, the MPEG pathway, the citrulline, and so on are metabolites. But these metabolites are not the same in the mixture. So this will create the so-called, the main process behind the response to the microplastic alone or the mixture. So the first one is energy metabolism, neurotoxicity, the AMP pathway, the protein homeostasis, and anti-inflammatory system. As you can see here, in the microplastic alone, the animal is expressing anti-inflammatory because plastic causes inflammation. Okay, so this is very important to depict and to understand. While in the second condition with the mixture of microplastic plus the herbicide, you have neurotoxicity the same, enhancement of cell apoptosis, so we are dealing with the cell death, energy, DNA metabolism, and inflammatory response the same. So we are some process that are the same between the two conditions, but we have here something very important, that the mixture of contaminant provoked the enhancement of the cell apoptosis. This is crucial. Okay, then the integration. Okay, I will escape and I will avoid to enter this very complicated schema, but at the end, what is the main message to take home, a take-home message? The first one, the result of the current study contributed to better understanding of the impact of co-exposure to microplastic and the herbicide. This is an evidence. The combined exposure to microplastic and the 2,4-D, the herbicide, had a clear effect on earth gut microbial shaping with change in gut microbial diversity. This is also evident. The study also identified significant changes in the metabolomic profile of earthworms. Finally, the study suggests that the impact of microplastic on earthworm gut microbial diversity depend on microplastic type and size, as we have mentioned this before, and the earthworm gut microbial community are closely related to the organism health and metabolism, since we are speaking about programmed cell death, anti-inflammatory response, and so on. So this is the last slide of this first part of the presentation, and it is my also I'm very glad also to say that this work has been recently, I mean, in 2024, in January, accepted for publication in the Journal of Hazard Material, with an impact factor of 14.22. So really this has been published in environmental pollution, yes, with 10 of impact factor, yes. No, the hazard material will become after. So this is our fantastic work, co-exposure to environmental microplastic and the pesticide that induce distinctive alteration in the metabolome and microbial community structure in the gut of earthworm Eisenia andrea. Part of this work has been done in collaboration with friends from the University Cattolica of Milan, so are not so far, in Piacenza. Okay? So probably at the end, we will have a session for question, but if you want to have some question now, please, the floor is yours. Otherwise, I will go further with another example. It's in the early life stage as a robust tool for ecotoxicological investigations.
Okay, I
So,
will ask if
as you like.
yeah, let's maybe ask if there are questions on this first part.
Yeah, Domenico, please.
Okay. So we saw like properly on real life how the assessment of microplastic and its effect are happening in real biology. I don't know if there are questions regarding the first part or curiosities even, okay?
Yeah, I would say curiosities more than questions, probably.
Okay, seems no, so we can go ahead.
Okay, good, go ahead. Okay, so the second part will be dedicated to the early life stage, a robust tool for ecotoxicological investigation. So in each life cycle, we have the early life stage, even for us humans, but also the adults. And there is a conflictual, let's say, situation that people prefer working with adults than the early life stage. From an ethical point of view, the early life stage are really an advantage for research, since we don't need some let's say permission or some ethical consideration, just because early life stages do not manifest the pain, so they do not feel the pain through the application of certain condition. So and they are really very, very vulnerable to some environmental conditions. So I will start with this magnificent element from the Mediterranean Sea, but also from all the world seas, Mytilus galloprovincialis. This was one of the best research that we have conducted in our lab, because it is directly linked to the environment and directly linked to the extinction of species. So in this study, we just evaluated the possibility of hybridization, and you will see then the impact of microplastic. But in the wildlife, you will have Mytilus galloprovincialis, which is more Mediterranean species, and Mytilus idylus, which is more Atlantic species. Unfortunately, with the climate change, the Mytilus galloprovincialis becomes to move or started moving toward the north space, and we have the so-called confluence sites. And one of the main confluence sites is the Gulf of Gascogne in North France, where the two species now are coexisting. So what is the consequence of the coexistence of two species? The coexistence of two species will lead to phenomena called the hybridization, and you will have the creation of hybrids. The problem with hybrids that you can have hybrid in the environment, but one of the most problem of hybrid that they could not reproduce. And in this case, probably we are facing one of the crucial episodes of species extinction, because if hybrid will be unable to reproduce, this species will go to nothing. I mean, so what we did here is just to obtain, and this is the life cycle of the Mytilus galloprovincialis, with this crucial phase which is called larv D or D larvae, because it has the shape of a D larvae. So we are able in the lab to obtain this D larvae and to conduct experiment on this very early life stage that occurs 48 hours post fecundation. Okay, so what we did here is very simple. We get And to obtain this gametes, I mean male or female gametes, it's very easy. Thermal shock up. You maintain your adult in 50 degree, then directly in 20 degree. And this thermal shock will provoke the emission of gamete. Of course, you need to have a mature gametes. So it is very easy, let's say relatively easy to make this in the lab. And then you will make the co-presence of male and female gametes to obtain the larvae. And then this larvae and this embryon, sorry, will be exposed to chemicals to obtain a 48 post-fecundation larvae that will be assessed for many aspects. So what we did here is very simple. Male and female Galloprovincialis, male and female Aedulus. So we will have a pure Galloprovincialis and a pure Aedulus. But in the same time, we will cross male Galloprovincialis with female Aedulus and male Aedulus with female Galloprovincialis to obtain hybrid from female Galloprovincialis and hybrid from female Aedulus. I think that it is more or less clear. So here are the first stages of development, and I want to show you, because this picture was taken in the lab. So this is the presence of the global, the first stage, then the first segmentation, then you have here something around 1, 2, 3, 4, 5, 6, 8 cells, but you can see the on the behind the floor. Yes. We applied here a very interesting technique after extraction of RNA called the microarray. And I hope that you know what is microarray. It is a dual color hybridization technique that will allow you to know the transcript in both situations and the so-called differentially expressed genes. So the technique, in one word, the technique will allow you to depict and to understand which are the gene which will be differentially expressed from one condition to another. So all these ones have been studied: Galloprovincialis larvae, Aedulus larvae, hybrid first condition larvae, and hybrid second condition larvae, and we have obtained this list of genes that are differentially expressed in animal exposed to 22 degree respect to animal exposed to 18 degree. So our challenge is the increase of 4 degree in the environment, which is the most realistic scenario that have been published by key scientists in Nature, saying that the Earth will know at least an increase of 4 degree in the next 15 years. Probably it will not be the case, but this is a scenario that have been proposed by the most important science in the world.
Okay. That was second.
Yes.
Yeah. And just to recall to one activity that we did last Friday, we went to the Civic Aquarium of Milan, and we were explained that the Mediterranean is gonna be tropicalized, no? So the increase of the temperatures, and actually what they assessed with this experiment is the effect of the increase of the temperature, and we saw that the increase of the temperature in this case has also modified the genetical expression of the.
And this is an example. Yes.
Okay.
This is an example.
Thank you. Just one curiosity, in the previous slide, you show some image at the microscope. Yes, yeah. This was like by the electronic microscope, right?
No. This is an inverted microscope with a magnification of 400, because the embryon of this organism is relatively big. So you can see it with an amplification of 400-fold. But you need an inverted microscope. So these are inverted microscope images. They are fantastic, really.
Okay. Thank you very much.
Okay. So yes, I will not, as I did for the first example, here are the output of the transcriptomic analysis. The main issue, as Domenico just said, is that there was a clear evidence of a change in the gene expression pattern of this organism when exposed to temperature increase of 4 degrees. But I will go directly, yes, this one. This is the second technique. This is another technique called the immunofluorescence. And in this technique, you can make a dye that will recognize main proteins inside the cell. And as you can see here, what has been, let's say, marked is the total larvae, the D larvae, okay, is the D larvae. So here we have the tubulin in red, the actin in green, and the merge of both of them, and also the merge with the DAPI. The DAPI is a dye that will recognize only nucleus, I mean DNA. So you can see here the DNA of the cells, very clear, and the repartition of tubulin and actin inside the D larvae, 48 hours after the fecundation. What is the main purpose of this dye or this approach? Here we were following the increase of tubulin inside the cell. What does that mean that the tubulin is increasing? Just to remind, the tubulin is a protein, a key protein in the cytoskeleton organization. The cytoskeleton in the cell, when damaged by the temperature, the cell will express more need to produce tubulin. So when the level of tubulin will increase in terms of presence of the protein, that means that there was an alteration of the tubulin, and that means that the cytoskeleton is injured. And when the cytoskeleton is injured, this is a drastic problem for the cell survival. So this was to prove that, as you can see here, that the tubulin level, we have depicted the histogram here, the tubulin level was maximum in Hedulus larvae. I mean, Hedulus larvae are really facing the drastic effect of the increase of temperature. So this first to prove that the larvae, the hybrid from a mother Hedulus, are not really very safe in presence of temperature increase when compared to Gallo provincialis.
Okay, just one second that I like these. So basically, just to summarize what they show, they analyze that the presence of this particular protein that is called tubulin. Tubulin is kind of a protein that is giving like the shape or the structure to the cell, no? The cell is a complex system and so on. But this tubulin protein is giving sort of infrastructure for the cell to be there, no? And the increase of temperature has increased also the production of this protein, and we can say the structure of the inner skeleton of the cell, as we can say, increased, has been more, we can say, prominent and so on.
Yeah,
correct. Yeah,
absolutely, yes. As a mitigation strategy, I mean, the protein in its native situation has been altered by the increase of temperature. So the cell will need the more production of this protein, but this will lead to a disorder anyway in the cell. It is well explained by, yes, like this, I think it is well explained. Yes, and this is the HRG. So here I will make a small stop. What is HRG? Histidine-rich glycoprotein. And I'm interlinking the cellular function with the, the total shape of an organism. The HRG is a protein, it is a calcium binding protein, and it is responsible of the deposit of calcium to the growth of the shell. I mean, the shell of this organism will grow year after year, and this growth is mainly due to the deposit of calcium. So the protein that will deposit this calcium is called HRG. It is the histidine-rich glycoprotein, and it is expressed genetically, and then the protein will make the work directly into the cell. So what we have observed here, that the level of shell malformation in increasing temperature is very high. And we try to explain why the shell formation is becoming so damaged when we increase the temperature. It is, and the answer is very clear here. And as you can see, the intensity of the green light here is due to the HRG. The intensity of the green light in Mytilus galloprovincialis and Mytilus edulis is not the same with pure edulis and hybrid from female edulis. And hybrid from female edulis are losing completely this green dye. What does that mean? That means that in hybrid from female edulis, we are losing the ability to deposit calcium. So that's why the shell is not well formed, and that's why we are facing a malformation situation, which will lead to the death of these early life stages. So compared with control larvae at 18 degrees, only pure galloprovincialis larvae showed an increase in HRG. Only pure galloprovincialis, because galloprovincialis are originated from sites from the Mediterranean, so they are accustomed with the increase of temperature. However, pure edulis and pure and hybrid from female edulis are not. And this was our main evidence that even the resistance, if it is transmitted, will be transmitted by female galloprovincialis, because edulis female are not able to transmit this so-called ability to face the increase of temperature. So this was the first evidence world ever that demonstrated why female pure edulis or hybrid from female edulis larvae cannot survive in high temperature. I mean, high temperature is 22 degrees, yeah? Okay, so the most part of the flourishes, yes, please.
No, just to summarize the discussion. So they saw that between the two species that gonna hybridize, only one can resist to the temperature, and the other one, you can say, is not developing such resistance. And even though we can say there are hybrids, the hybrids are like not so strong, we can say, as the species that is transmitting the resistance. Of course, this creating some we can say disequilibrium into the environment, and also of course we can say that the increase of temperature is damaging in this case, we can say the more autochthonous species, right, Mohammed?
Yes, absolutely yes. I mean, this slide shows very well how can an invasive species, because Mytilus galloprovincialis is considered in this case invader, because she's invading the northern part of the Europe. It is originated from the South Mediterranean coast and from the Mediterranean shore in general, where the temperature is in the most time around 20 degrees. But in the North Mediterranean area, I mean in France, in Norway, in these regions, the temperature can reach 12 degrees. But now with the climate change, the temperature is increasing day after day. And this provided in somehow a new ecological situation in favor of Mytilus galloprovincialis. And this is the first evidence why galloprovincialis survived in this area, but not edulis. The native species could not survive in a changing climate because they cannot produce the so-called protein of deposition of calcium that will ensure the formation of the shell.
Okay. This is, for example, we can make a comparison with this situation with the one that also we saw into the aquarium, where, for example, clownfish were coming into the Euro Mediterranean, but they were belonging to the, we can say, to the Red Sea. But indeed, other species, and we saw several of them were disappearing, probably for the fact that the temperature was different. In this case, you see the temperature is affecting the creation of the shell of the mussel. Yeah.
Yeah, absolutely yes. So I mean, if you would study the effect of temperature on adults, you will not see this because they are already with a well-formed shell. But when you look to the early life stage from the beginning of the history, I mean, of this organism, you will see how important is the formation of the shell, because an organism without a shell well formed will die. And the temperature is a key factor in the production of the shell. And now we are also seeing the same situation with microplastic. That's why I started with the temperature, but I'm arriving to microplastic, since microplastic is interfering with the shell formation. Okay, so I will go also further. And this is a fantastic also illustration that we had in the lab called the calcium deposit with alizarin red staining. The alizarin red staining would recognize the calcium in one tissue. So as you can see here, from the control situation, the red is really, I mean, the pink is very intense because calcium is there. But when you test your larvae with DMSO, with microplastic, with 100 microgram liter, with diuron, which is an herbicide, and the mix of diuron and microplastic, you see how how contaminant affect the deposit of calcium. So when you combine the two approaches, the level of expression of the histidine-rich glycoprotein, the protein that have to make this accumulation of calcium, and the accumulation of calcium itself, you have idea very clear. I mean, the protein is no more functioning and there is no deposition of calcium. So it's clear for us. The situation is really very clear. Let's move now to another situation already, also with mussels. And this is a very tricky one. I mean, I'm really very proud about this publication because the level was really very high. The idea is original. I mean, we took the spermatozoa of the mussels and we exposed them to 1, 10, 15, 100 microgram per liter of microplastic under a continuous shaker of an hour. And then we tried to analyze what is happening inside the spermatozoa physiology. And why we did this? Because in contradiction with humans, these organisms will release the spermatozoa into the sea, and the female will release the oocyte into the sea. So both gametes will encounter themselves into a contaminated environment. However, for us, I mean mammalians, the male will deposit directly the spermatozoa into, let's say, a protected area. Okay, and this protected area will ensure at least one gamete will make the fecundation. However, in the open sea, you will have a situation where the spermatozoa and the ovules will be in probably a high contaminated area. So here, the question is, will microplastic affect the stability and viability of the spermatozoa? And why the shaker? The shaker because when these two organisms will release, it's called the spawning, release the gamete, it will be in a, let's say, a zone where the waves will play a role. So it is a continuous shaker also. So what we analyzed here is very simple, the DNA integrity after 1 hour of exposure. And as you can see, using the acridine orange test, the DNA integrity was lost. I mean, acridine orange positive spermatozoa means these spermatozoa are presenting an altered DNA. The DNA was completely altered after exposure to microplastic. Then further, sorry, I haven't the data dealing because we established it also here that the integrity of DNA was altered because we depicted a phenomena of apoptosis that is manifested by increase of BAX and decrease of BCL2 proteins and of cytochrome C release also. So it is a physiological study. So why I'm proud of this, because this has been published on Journal of Hazard Material with an impact factor of 1422. And the paper is unveiling the impact of environmental microplastic on mussel spermatozoa. So in this paper, we also provided the first evidence of protein alpha detection in invertebrates. Protein alpha, for those who do not know this, it is very well known in humans and invertebrates in general. And this protein is the one responsible of the penetration of the spermatozoa into the ovule. Okay? So if there is a lack of this protein, the spermatozoa would not be able to penetrate and to make the fecundation. Yes, let's move to this fantastic image. Fantastic and very amazing. Why? Because these are larvae of sea bass, a very common fish. And we have in Tunisia a very important society, private society that produces larvae, and then this larvae will be sold to make the the maintenance and to obtain sea bar of white of at least 500 grams. So the first life stage are these ones. These are larvae of 6 days post hatching. And as you can see here, you have this microplastic inside. Microplastic that will conditionate the future of this larvae, because if this microplastic will block the anal orifice here, this will lead to the death of the animal. Otherwise, they will escape. Yes.
Just 1 second. I want to highlight one thing. Also,
Yeah.
taking the previous example of mussels. First of all, we saw with the mussels the importance of the developmental stage and the age of in which the organism is exposed. No, when we were
Yes.
like talking about toxicity the last time, one of the factor into the toxicity study was like the age of the organisms that were exposed and also the developmental stage. If they are developed like exposed into the, we can say, reproductive stage, it's very risky, okay, because they are going to affect the fertility of the species.
Yeah.
And also this thank you for bringing this last example, because why to study the toxicity of microplastic and so on and so forth. This was like valid for this example, but I can say that, for example, some type of mussels, they are also, we can say, a problem for human communities regarding, for example, the food chain. No,
Yeah.
in the Mediterranean, we are very used to cook with mussels, and also there are the fishermen that are taking the mussel also for an economical purpose. No. And so it's not that much surely for the ecosystem, that's the most important, but sometimes these effects are gonna affect also the human activities.
Absolutely yes. Absolutely yes. Okay,
Thank you.
so I will move also. I will not make a focus, but just to let you know that we have also published a very good paper related to the microplastic and interaction with the mussels, but not only mussels. There is also a very nice publication that deals with the bacterial biofilm that could be formed on floating microplastic. And this picture, this was the graphical abstract of that paper called the Bacterial Community Profiling of Floating Microplastic of the South Mediterranean Sites, in which we report the presence for the first time of pathogenic bacteria that normally do not exist in the sea, but they could fight the salinity being associated to microplastic. In this case, it was Vibrio fischeri. So this is very tricky in the marine environment. Yes, I would skip also this part of the presentation, because it is also very important to highlight the interaction between antibiotic and microplastic in the production of chicken, since we know very well that in chicken we are using, for the production of chicken and for the farmers that work with chicken, they use a lot of antibiotics to maintain them. But in the meantime, chicken can also be exposed to microplastic through the water that can drink, because they are really, the situation with the rearing activities is promoting in somehow the use of plastic as let's say, devices in which the chicken can drink. And as you can see, as you know for sure, that chicken, when they are drinking, they are also creating some alteration in the plastic with their I don't know the term, but yes, microplastic can be present in their drinking water. So the idea in this paper is to evaluate wherever microplastic can contribute to the accumulation of antibiotic in chicken liver, and how far this could be harmful for chicken. And I would go very fast because my time is running out, but yes, this is crucial because these two publications showed that micro and nanoplastic in edible fruit and vegetable. We did these two publications with colleagues from Catania, Professor Margherita Ferranti and Gea Oliveri Conti, when we reported for the first time the presence of microplastic in fruit and vegetables, and also in edible part of the Mediterranean fish. And I can say, because I conducted the study for the Mediterranean fish, we used the sea bar, solula, and also trilia, and also mussels. So we know very well that from a very typical Mediterranean diet based on vegetables, but also in seafood, we can be contaminated ourselves by microplastics. So the question now, wherever microplastic can induce alteration in humans, and I will conclude with this. So we tried to study the effect of microplastic in human mesenchymal stem cells, two types of mesenchymal stem cells, the one from the bone marrow and the one from the adipocyte. So the idea is to make the to have these two kind of cells, the bone marrow and the adipocyte. They are stem cells, and to make the exposure of these two stem cells to microplastic that we have generated from what bottle of water, I mean polyethylene terephthalate. We just frozen the bottle of water in liquid nitrogen and then ground them and then filter them on a filter of 2.6 to obtain microplastic less than 2.6, then less than 1 micrometer. So we have two range of sizes, the one between 2.6 and 1 micrometer, and the one under 1 micrometer. And then we made the exposure to these two kind of stem cells, human stem cells. So the idea is to evaluate first the cell division. As you can see here, from the adipocyte or the bone marrow stem cells, we have a crucial reduction or a crucial stop of cell division. And this is really very worrying. I mean, human stem cells that should increase their division in an exponential way, when exposed to microplastic, we have a reduction of the division. So in certain how, microplastic will provoke the arrest of cell cycle. So this was also identified using the so-called cell cycle analysis. You know very well that in the cell cycle we can have the phase G1, G2, and S. And here we have obtained evidence that the percentage of cells in S phase decreased in adipocytes and bone marrow after treatment with polyethylene and polyethylene terephthalate, with the two types, I mean the two dimensions. So we have a reduction in the phase of DNA synthesis. The DNA synthesis phase decreased drastically. So this is in correlation with the cell cycle, I mean the division of these cells. Yes, I will escape this to go to more, yes, apoptosis. And we found also that apoptotic cells that have been studied through flux cytometry, yes, cytometry or flux cytometry. And if you want, I can provide 2 words about this technique. I don't know if I have to do the work of
Please do. Was also
okay.
one of the techniques that we addressed in the
So this technique of flux cytometry is a technique when you can make a special dyeing for cells that are undergoing one pathway. I mean, with a special dye who will recognize a specific protein in the cell, and then you will quantify the intensity of the fluorescence that is that is corresponding to the process that is undergoing by the cells. So in this case, for apoptosis, we are dying a specific protein of apoptosis. So when we have an increase in the shape, in the intensity, that means that these cells are undergoing apoptosis, and these are the shape of the image that we obtain. These are images that are obtained after flux cytometry analysis.
Okay, just 1 second. When we say apoptosis, that means that we say the death of the cell. Okay.
Yes, yes. Probably I also need to say 2 words. The cell can die through 3 processes. One is the necrosis. The necrosis is an imminent death. I mean, you will take your cells and you will put them in the oven, and then you will turn off the oven. And in 5 seconds, you will have a death of the cells. But it is a sudden death of the cells. No process are engaged. These called the necrosis. Then you have apoptosis. The apoptosis is a programmed cell death that is controlled, and the main protein of apoptosis, the first one and the major one, who is, let's say, provoking this, is called the caspase. The caspase is the protein which executes the cell death. But of course, before the execution, there is a path that could implicate the Bax, the BCL2 from the mitochondrial, but also the cytochrome C and so on. The third cell death, programmed cell death, is called autophagy. And the autophagy is also a programmed cell death under the control of main proteins. One of the main proteins that are controlling autophagy is called the mTOR, mammalian target of rampicin, but also LC3, LC2, and P62. Well, the autophagy is a kind of cell death that will be provoked by the increase in the dimension of the lysosomes. And in this case, the lysosomes will make the auto-invagination of the cell. However, the apoptosis will start with the degradation of the DNA. So there are 2 kinds of cell death, but both of them are programmed cell death. However, the necrosis, the third cell death, is a sudden and very quick cell death. Okay, so yes, let me show you. Yes, this one is very important. I mean, we also try to understand which will be the future of these cells exposed to microplastic. I mean, bone marrow mesenchymal stem cells, when exposed to microplastic, should provide at the end 3 types of cells. The first one is called adipocytes in a very minimal amount, chondriocyte, and osteoblasts. Chondriocytes will also turn into osteoblasts at the end. So these cells would give to the body the core and the main source of bones. However, it could also give in somehow some fat through the adipocyte. What we have obtained in our results, that the exposure to microplastic not only reduced the division, not only increased the programmed cell death, but mainly shifted the future of the cell differentiation into adipocytes. So in somehow the microplastic in this case is provoking and is causing a state of fatness. I mean, people can become very fat with an extra accumulation of fat due to the production of adipocytes. Of course, this is a cellular scale and could not be translated to a total human body, but this is the main idea.
So just 1 second.
Yes.
I like this. So basically, the presence of microplastic is gonna alter the it's called differentiation of the
Yes.
osteoblasts, that instead of doing bones, they are doing fat cells. Okay.
Yeah. And the problem is, up now, we are not really convinced how this could be happen. That's why we are collaborating with Professor Izzotti from Genova, who is doing some mRNA analysis on the same cells we used for this experiment to try to find out key processes that are damaged, altered, and that shifted the cell differentiation into adipocyte. My last slide would be this one: analysis of damaged DNA by X2AX and 8AM. These two techniques. One is key regulator of cell response to DNA damage, and the ATM is a kinase that regulates DNA repair. So both of them are acting to regulate cell response to DNA damage and to how to recover the DNA. So as you can see also here, the two main proteins are overexpressed. That means that DNA is altered in this case. So DNA was altered, that's why the cell is expressing key protein of cell reparation and cell repair. Yes, I would stop with this. I have now 1 hour 30 minutes of, and I would like, as agreed with your professor, to give you the floor for 10 minutes of reflection, 10 minutes of question, probably of curiosity, as supposed before, about this presentation. Thanking you very much for your for following me and for your interest. So thank you very much.
Thank you to you, and congratulations for your great work. So yeah, now if there are
I will stop the share probably.
okay.
I will stop the share.
Okay.
Okay. Now just Domenico to give them the floor to ask questions, to have some curiosity and so on.
Okay. Thank you. So the floor is to you if you have some curiosities also. I mean, we saw very in deep work that is happening for the characterization of the microplastic risk. And one thing that I want to highlight that catch a little bit my attention, for example, when they were studying the muscle reproduction, they tried to, you can say, reproduct the same conditions also in terms of flows. No, they use like the mixture properly to reproduce the same condition. That's very important into the so-called scientific methods and scientific methodology. Okay, into the lab you are trying to simplify the condition, but to reproduce the one that can in a way affect the phenomenon.
Absolutely yes. Absolutely yes. And actually, we are conducting a new research to find out solution of mitigation of plastic once in the cell. I mean, we can face the problem of the plastic, saying how to reduce plastic into the environment. But I'm convinced that the only one solution is to stop using the plastic at its at this level, but this would not be possible in view of the political, of the economical considerations. So probably we need to face the problem in another way, trying to find solution to make robust this response at the cellular level. And we are testing now the effect of melatonin, for example, to mitigate the microplastic effect at cellular level. This is a fantastic work that we are conducting on human stem cells. We are also now in an ongoing experiment studying the metabolomic and the metagenomic profile in fish early life stage gut to understand what is happening in fish. We are also doing the same with earthworms, with seaworms. Anyway, we are trying to find solutions to this crucial problem, but not only microplastic that is depicting our attention. Now we are working also with PFOS, one of the main killer in the environment. Unfortunately, and even the PFOS have been stopped of use after an European decree. The problem that we are in a very high level PFOS in the soils. So we need to find solutions, probably to find some key bacteria that could degrade the PFOS. But this would be also through the investigation of earthworms, because in the gut of earthworm, we have a mine of bacterial information. So probably also there we are working very hard in collaboration with other colleagues and so on. Yes.
Thank you. Thank you very much for highlighting this point. We are telling several times in this course that we are trying to understand the problem, but also to find a possible solution. And that's important also to study this kind of phenomena. Okay.
Yeah.
So, yeah, please, if there are questions. Yeah, yeah. Yeah. So the question is, so the work that you are doing into the Mediterranean can lead also to some consideration, assumption for other seas, and so how to deal, for example, with the phenomenon that can happen in other seas, and why Mediterranean is so special.
Okay, thank you for this question. First, the Mediterranean is special because we are on the Mediterranean, so we are working in a context of common sea between the north part and the south part of the Mediterranean. The Mediterranean is very special because it is the smallest living sea in the world. So we are facing really a very crucial problem with the Mediterranean in terms of contamination. But it is not the only, let's say, experience we get. We worked with microplastic from the Caribbean, microplastic in Hawaii, and we receive in continuous samples from all over the world. From the Atlantic, we are working with a group from Agadir in Morocco, from the Atlantic Sea, and we have published recently a comparison between the pollution of microplastic in the coast of Morocco, the Atlantic coast, and also the Mediterranean coast of Tunisia. And it was really very interesting as work. And since you are talking about the other seas, one of the main results that we obtained along with our colleague Professor Jérôme Cachot from Bordeaux is from the Caribbean, and he obtained in this investigation, Professor Cachot, that on microplastic he found some trace of DDT. It is an insecticide that has been forbidden from production, from use for more than 40 years. And we can found until now some residue of DDT on microplastic in the Caribbean. So this is really very frightening because microplastic can be, under the water flow and the currentology of the seas, we can have some plastic from the US that enter the Mediterranean, some plastic from Italy that can arrive to Tunisia, and so on. So the plastic is not a problem of one country. It is not a problem of one sea. It is a global problem, and we have to face it all together.
Thank you. Thank you very much. One point on this, we will see also in the second part. Currently, as Professor Banni was saying, it's important that plastic is addressed at the global scale. During these days, in Nairobi, it's happening the UNEA 6, that is the UN Environmental Assembly. Is basically the main assembly of the Ministry of Environment and so on under the coordination of UNEP, the UN Environmental Protection Agency. And one of the elements into the agenda is probably the so-called plastic treaty. So a global treaty to stop the production of plastic. We will see something about that in the second part of the course. But just to say that it's important that, as Professor Banni has said, this problem has no borders. So everyone needs to be involved, and there is the need of a global solution first.
Yeah. I will just add the one word, since we have been granted this year an Horizon Europe project dealing with the plastic and packaging. I mean, how can we reduce the migration of plastic from the packaging to the food? And we are really starting this fantastic project called Magno. You can MAGNO. You can find it online, and the Horizon Europe funded project. And probably we will get also more financial support to study this phenomena.
Thank you. Thank you very much. There is space for one last question. eventually, if there are. Okay. So if there aren't, so Professor Banni, thank you very
Thank you
much.
very much.
It's been a real pleasure, and really
Pleasure is
congratulations
mine. Pleasure is mine.
For your work. And yeah, if you have questions eventually after the lesson for Professor Banni, just like write to me, and I will try to forward to
I will
him.
respond via email to all kind of questions. Don't worry about that, please.
Thank you. So have a nice day, and
Thank
thank
you
you very
very
much
much.
again.
Have a nice day in Milan. Thank you very much. Ciao,
Bye
ciao.
bye. Bye
Ciao,
bye
ciao.
bye.